Hi, my name is Yue Wang. I'm from Tsinghua University. Today, my topic is a single-cell proteomic snapshot of early T-cell development in human infant thymus. T-cells play important roles in human adaptive immunity. Studying protein expression patterns during early T-cell development is crucial for understanding their differentiation process and function mechanism. So our research goals are to infer a human thymic T-cell developmental trajectory using single-cell mass cytometry data to find out cell types, to observe protein marker patterns, to identify defining markers for certain cell types. Here is an overview of methods. The first step is sample collection. Thymus is fully formed at birth. It will shrink as we grow up. Therefore, all major subtypes of T cell appear in the thymus during the infant period. So we collect 5 thymic samples from human infants of 1 month to 1 year old. Then protein expression levels of each cell in the samples were measured by mass cytometry. We designed these marker panels with protein markers, including cell surface markers, transcription factors, and phosphorylated transcription factors. They were measured by mass cytometry. As we got our data, we conduct pre-processing with gating and arcsine age transformation. Here are the gating results. In the figure, each dot is a cell and color represents intensity. We change to different parameters to select certain cells we want. For example, gating on DNA channels was used to remove debris. Gating on cisplatin to select variable cells. Gating on event length to make sure it's single cells. This one is quality control, and gating on CD45 to get immune cells, to get rid of dendritic cells, to remove epithelial cells, and remove B cells, and only left with T cells for the downstream analysis. Flowosomes was used for downstream analysis with following steps. First, clustering with self-organizing map, and then build minimal spanning tree on the cluster results so we can observe protein patterns on the tree. Lastly, we assign pseudotime to each cluster of certain branch on the tree and observe the marker trend. Here are the flowosome results for all the samples. Each node is a cluster, and each color represents a cell types. Those are the cell types we identified. From the flowosome results, we generate this graph. In the graph, we have proteins that we use to define cell types, and we show the whole developmental process on this figure. So the process starts from pro-T cells, and it will develop into different T cell branches and further differentiate into more functional cells. For example, regulatory T cells, in short Tregs, T helper 17 cells, and etc. Here are the protein expression patterns throughout the T cell developmental tree. So medium protein patterns of different markers was shown their upregulation or downregulation in different stages. For example, Ki67 and CD73 and Helios, they showed upregulation in the early CD4 positive, CD8 positive branch. And other markers, including phosphorylated transcription factor. They showed upregulations in different stages of the development. Clusters of the developmental tree in this red line were assigned with pseudotime from the beginning cluster Prote to the terminal Treg cluster. So for example, Prote is assigned with pseudotime 1, and here's 2, 3, and till the end is 20. The patterns of Treg relevant protein markers were plotted with this line graph in respect to pseudotime, so we can observe the trend throughout the development and find out the defining markers for Tregs. In conclusion, our work showed a human thymic T cell developmental trajectory from single cell data, and we showed a demonstration of T cell subtypes and observed protein marker patterns and find out the key markers for certain cells such as Treg. And our work provide an analysis framework of cell developmental trajectory inference with human cellular proteins. That's it. Thank you very much.